Hey everyone, welcome back to another videos. So in today's video I'll be talking about mutation and I'll be focusing on chromosomal abnormalities. So first of all, what is mutation? Mutation is a few changes that occur in the genetic material of the organism. These changes occur mostly unexpected and random as well. So as a result, either a gene or complete chromosome gets affected. So the two major types of mutations are genetic mutations and chromosomal mutation. As I said before, in this video I'll be talking about chromosomal mutations because in the next video I'll be talking about single gene mutation by taking also a few examples to make it more clear for you guys. So before talking about mutation and how can occur in the organism, in the human organism, let me explain a chromosome. So chromosome is are divided by two regions, which is called arms, which referred P for petit arms, which is the short arm, Q, which is referred for the long arm, and are linked by a centromere that link the two sister chromatids, and the telomere, which is present at the near end of each chromosome and contain repetitive nucleotides sequences. So as I said, the centromere play a lot of roles, such as linking between the two sister chromatids, and also it depends on the position of the centromere which type of chromosome can be, because it can include metacentric, submetacentric, acrocentric, and telocentric. And start talking about metacentric as well, as we're seeing in the picture. Metacentric is when the centromere is placed almost exactly at the center. The two arms seems almost at the same length, so the centromere is placed in the middle of the chromosome structure. Submetacentric is when the centromere is placed slightly away from the center, so the two arms of the chromosome appear unequal. The next type is the acrocentric. The centromere seems almost towards the end of regions. The human acrocentric are 13, 14, 15, 21, and 22. Telocentric, which is the last type of chromosome, is present at the extreme end of the chromosome, and these chromosomes are not found in the human so there's a lack in the human karyotype of this type of chromosome. And as we know, the human karyotype are composed by 46 chromosomes, which are divided between 22 pairs of autosome chromosome and sex chromosome. And when I'm talking about autosome chromosome, which is between the first and the 22 pairs of chromosome, the last pair is the sex chromosome. And when we have XX, which determine a female, and XY, which is referred to a male as well. So talking about chromosomal mutation, they can be occurred by two ways: disruption in the chromosomal structure or disruption in the number of chromosomes. Talking about the first one, which is the aberration of the structure of the chromosome, can be caused by so many different ways, such as deletion, which is loss of segment of the genetic material, and it can be happened by terminal deletion or intercalary deletion. Terminal deletion, when the deletion or the loss of the segment of the chromosome is near one end of the chromosome. Instead, intercalary, which is the deletion is occur in the middle of the chromosome. Talking about duplication, when we have an extra or it happened that there's presence of one extra segment of the genetic material. Insertion, when there's added part or a segment from another chromosome. Inversion, which is an other chromosomal abnormalities, and in which, in order of genes, the segment of chromosome can be reversed as 180 degree, an angle of 180 degree, and it can be pericentric or paracentric, in the sense that paracentric, the inverted segment is occurred on the short arm of the chromosome or in the long arm of the chromosome. Instead, in the pericentric inversion, the inverted segments include the centromere, so it's around the centromere and it's between the short and the long arm of the chromosome. Talking about another chromosomal abnormalities, which is the translocation, when we have an interchange of a segment of movement of a portion between one chromosome to another, and when we have an interchange of chromosome between two non-homologous chromosomes, we're talking about reciprocal translocation. And the translocation can be balanced or imbalanced as well. So we have a most common translocation, which is Robertsonian translocation, that occur in the acrocentric chromosome. Acrocentric chromosome that, as I mentioned before, are 13, 14, 15, 21 and 22. So the person that carries a Robertsonian translocation, he's normal and he's asymptomatic as well, but can produce gametes that are imbalanced, which lead to have a trisomic or monosomic zygotes. Other chromosomal rearrangement are ring chromosome, when two undergoes breaks in a chromosome and the end of the chromosome will reunite to form a ring structure. And isochromosome, which when in a chromosome we have an arm of the chromosome is missing and the other arm is duplicate in a mirror fashion image. But these two types of chromosome are rare as well. Moving on, I would like to talk about chromosomal numerical abnormalities. The most common chromosomal abnormality, which is called aneuploidy. Aneuploidy, which is due to a missing or an extra copy of a chromosome. And the most common aneuploidy is been seen as a trisomy, when we have a 3 copy of chromosome, or monosomy, when we have a single copy of chromosome. The most well-known aneuploidy, which is Down syndrome, when we have a 3 copy of the chromosome 21. For this reason, I prefer to do another video talking in details about Down syndrome. So other trisomies has been seen, such as Edwards syndrome, trisomy 18, and Pato syndrome, trisomy 13. Also, aneuploidies has been seen in sex chromosome, such as Klinefelter, when we have an extra X copy of the chromosome, 47XXY, and also the only monosomy, the viable only monosomy, which is 45 and only one single X chromosome, which is related to Turner syndrome. And also another type of abnormal chromosomal mutation, which is polyploidy. Polyploidy is been found in the human cells. It's rare as a condition, but it can be triploidy, in the sense when we have an extra set of chromosome. Instead of having 2N, which is related to 46 chromosome, we have 3N, 3 sets of chromosome, so we have 69 chromosome, XXXX, and or tetraploidy, which is 4N, which is 4 sets of chromosome, and we have 92 chromosome, and it has been shown, the literature, that 70% of the abortion has been caused by polyploidy, in specific triploidy cases, and it can be cause a lot of problems, triploidy syndromes. So the main causes of polyploidy include fertilization with diploid spermatocyte, or fertilization with a single normal egg with 2 sperms. And it has been shown that most all of the cases of polyploidy are lethal as well. 90% of live birth died within 24 hours. Only mosaic cases can survive. Talking about mosaicism or mosaic cases, which is considered also as an numerical chromosomal abnormalities, because in the person with mosaicism can have 2 or more genetically different sets of cells, in the sense that he might have 46 chromosomes in some cells, and in other cells might have 47 chromosomes, which lead him to have problems in his body. If you made it to the end of the video, thank you so much. I hope you enjoyed the video, and I hope you find it helpful as well. Stay safe.